Hello student, hope you are fine. Myself Sri K.D. Borsesar, a biology subject teacher. In last lecture we detailed discuss embryonic development, different events or step involved during the embryonic development from zygote up to 3 germinal layers, cleavages, blastulation, implantation. Now today we detail discuss embryonic development, different events or step involved during the embryonic development from zygote up to 3 germinal layers, gastrulation. So let us start. Embryonic development, embryonic development from zygote up to 3 germinal layers, different event or steps involves like cleavages, blastulation, implantation and gastrulation. So above all three steps or events we detail discuss in previous lecture. So we today's lecture detail discuss about gastrulation. Before gastrulation, understand cleavage, blastulation, implantation, seen in basically what is actually cleavages. Zygote multicellular cell undergoes mitotic division to form 2-cell stage cleavage, undergoes mitosis to form 4-cell cleavage, undergoes mitosis to form 8-cell cleavage, and undergoes mitosis to form 16-cell or 32-cell stage cleavage called as morula. Each one cleavage cell called as blastomere. So morula is a solid ball like structure. So this morula reaches the isthmus. Isthmus is the part of oviduct. So this is the isthmus part, oviduct, distal part open into uterus. So in ampulla, fertilization process takes place. After fertilization, zygote is formed. Zygote undergoes different stages of cleavages. So morula is last stage of cleavage, is reaches up to isthmus. The morula is converted into blastocyst during the blastulation. So up to this here cleavages processes, morula reaches the isthmus. In blastulation, formation of hollow fluid filled multicellular structure, blastula or blastocyst are developed from morula called as blastulation. Morula structure undergoes development to form hollow fluid filled cavity structure, multicellular hollow fluid filled cavity multicellular structure, blastulation. Now seen, this is the blastocyst is developed from morula. Such blastocyst is end of 7th day fully formed, now ready for implantation. Up to fully formed blastocyst with present zona pellucida. This is the zona pellucida. Function prevent implantation to embryo at an abnormal site. When zona pellucida ruptured from blastocyst stage, implantation process occurs. Now during implantation, attachment or embedded blastocyst into endometrium of uterus. Such process called as implantation. You know, here seen morula stage is converted into blastocyst. So such blastocyst enter into the uterus. Inside uterus, blastocyst attached, embedded into the endometrium layer of uterus. Such process called as implantation. Here seen in detail implantation. This is the blastula is attached or embedded in the uterus lining of endometrium. So this is the process of implantation. This process is usually begins 7th day after fertilization and 10th day end. Embryo is completely buried inside the endometrium. Embryo is usually implanted in fundus region of uterus. So up to this we detail discuss previous lecture. Now we detail study, understand gastrulation. What is actual gastrulation? It is the process of formation of gastrula from blastocyst called as gastrulation. In simply, formation of gastrula from blastocyst or blastula, such process called as gastrulation. So this is the blastocyst scene. Blastocyst is undergoes develop to form gastrula structure. Vs of late gastrula structure seen here. This one is endoderm, this is the ectoderm, and this in between ectoderm and endoderm, mesoderm seen. Each one contain cavities seen. In endoderm present yolk sac cavity, ectoderm contain amniotic cavity, mesoderm contains coelom, and on the coelom covering chorion. So this is the structure of gastrula have seen. Ectoderm, mesoderm and endoderm is developed from blastocyst. So we discuss how gastrula develop from blastocyst or blastula. During gastrulation, two important events takes place. One is differentiation of blastomeres and other is morphogenetic movement. So we seen one by one. First one is differentiation of blastomeres. This process result in formation of three germinal layers, that is ectoderm, mesoderm and endoderm, from cells of embryoblast. You know embryoblast, it is present in blastocyst. So cells of embryoblast from develop germinal layers like ectoderm, mesoderm and endoderm is in differentiation of blastomere event. Morphogenetic movement, these are different type of movements to reach their definite place in embryo. So these two are most important event during the gastrulation. Now how gastrulation process occurs? Gastrulation begins in embryoblast cell on about 8th day after fertilization. So here noted point, gastrulation in the embryoblast cell on about 8th day after fertilization begins. Free end of inner cell mass or embryoblast, embryoblast or cell mass, inner cell mass are present in blastocyst. So free end inner cell mass, embryoblast called as hypoblast. They are primitive endoderm cells. Why hypoblast are called as primitive endoderm cell? Because endoderm is developed from hypoblast. So hypoblast primitive endodermal cell becomes flattened, start dividing and grow downward towards the blastocele cavity of blastocyst. Let me show with the help of diagram here. This is the endometrium layer and this one is blastocyst, trophoblast outer covering inside it. This is the blastocele cavity and inner cell mass called as embryoblast. Now these cells are free end cells of embryoblast. So free end cells of inner cell mass or embryoblast called as hypoblast. So these are the hypoblast cells. Primitive endoderm cells becomes flattened, start dividing and grow downward towards the blastocele cavity of the blastocyst. Seen here, these red color cells are hypoblast cells, start dividing and grow inside the blastocele cavity of the blastocyst. So this process is continuous to form endoderm layer. So this is the endoderm layer. Due to continuous dividing of hypoblast cell grow downward toward the blastocele cavity of blastocyst and to form endoderm. This layer called as endoderm, and this is the first differentiate. It grow within the blastocele and form sac called as yolk sac. So inside the endoderm present a cavity or sac called as yolk sac. Labeled them. This is the yolk sac. This one is yolk sac present in endoderm cells. So this is the cavity of endoderm. Such endoderm is formed from hypoblast cells present in blastocyst. So up to this we detail discuss how form endoderm layer. In simply, hypoblast primitive endodermal cells develop endoderm layer, and inside it cavity called as yolk sac. Now the remaining cells of the inner cell mass embryoblast in the contact with cells of a robber are called as epiblasts, also called as primary ectodermal cell. Why epiblast cells are called as primary ectodermal cell? Because from epiblast cells develop endoderm, so called as primary ectodermal cells. Now here seen, no doubt this is the endometrium and the implanted blastocyst have seen this. These are the hypoblast cells, and these cells are attached to trophoblast cell, robber cell. So the cells of epiblast of inner cell mass or embryoblast seen here. This epiblast cell from develop ectoderm. So this is the ectoderm developed from epiblast cells. Here seen both layers completely developed. You know, endoderm is developed from hypoblast cells, and here seen ectoderm is developed from epiblast cells. Now, how form endoderm? Cells of epiblast divide and redivide and move in such a way that they enclose the amniotic cavity. This layer to differentiate in is the ectoderm. So this epiblast cell form ectoderm, and inside the ectoderm seen amniotic sac or amniotic cavity. So here we seen how form ectoderm from epiblast. So inside the ectoderm present amniotic cavity is also seen here. So here up to this we understand development of endoderm layer, inside it cavity called as yolk sac, development of ectoderm, inside it cavity called as amniotic sac or amniotic cavity. So both layers form a flat bilaminar embryonal disc, seen here diagrammatic representation. This is the ectodermal layer and this is the endodermal layer. In both layer has form embryonic disc, seen this is the embryonic disc labeled. How form embryonic disc? Both layer form a flat bilaminar embryonal disc. So up to this we seen ectoderm formation, endoderm formation and bilaminar embryonal disc formation. Now the floor of this cavity has embryonal disc. Here seen the floor of cavity. This is the amniotic cavity and this one is yolk sac cavity and this is the embryonal disc. The pyramidal cells of disc towards the amniotic cavity form embryonal ectoderm, seen here amniotic cavity. The roof of amniotic cavity is lined by aminogenic cells. Here seen the roof of amniotic cavity is lined by aminogenic cells. Later these cells divide and redivide to form amnion. How form amnion? Seen here detail. The roof of amniotic cavity contains cells are called as aminogenic cell. Later this cell divide redivide to form amnion, here labeled amnion. So here seen how form amnion. Amnion is an extra embryonic membrane that surround and protect the embryo. So it is most important. As a result, all these changes, the bilaminar embryonic disc is positioned in between amniotic cavity and yolk sac. So here seen embryonic disc is bilaminar disc present in between amniotic cavity and yolk sac. So up to this we detail discuss here how form bilaminar embryonic disc present in between amniotic cavity and yolk sac. Actual gastrulation occurs about 15 days after fertilization. This is the noted point. Actual gastrulation occurs 15 days after fertilization, in which the bilaminar embryonic disc is transformed into trilaminar embryonic disc. Seen here diagrammatic representation, bilaminar embryonic disc. Here seen bilaminar embryonic disc. So this disc is present in between amniotic cavity and yolk sac cavity. So bilaminar amniotic disc is transformed into trilaminar embryonic disc. Here seen epiblast cell, blue colored layer. You know this layer is ectoderm, and hypoblast cells present. So this layer called as endodermal cell layer. Here seen primitive streak developed bilaminar disc. Here seen primitive disc is emerged in the both layers. Now the transformation occurs by division of rearrangement and migration of cells epiblast. Due to this formation of primitive streak and shallow groove on the surface called as primitive groove. So here seen transformation occurs by division of rearrangement or migration of epiblast cells. So these are the epiblast cells migrate, and all these epiblast cells are present in between outer ectoderm layer, and this one is endoderm layer. So the primitive streak and shallow groove seen on the surface called as primitive groove, seen here in detail. Now primitive streak progresses from posterior to anterior end of embryo. From side of primitive streak, a third layer of cells called as mesoderm extend between ectoderm and endoderm. Seen here diagrammatic representation. This is the ectodermal layer and this one is endodermal layer. This is the primitive streak and primitive streak location epiblast cell rearranged, migrate, present in between these two layers, and these cells are called as mesodermal cells. Here also seen ectoderm, and this one is endoderm, and these cells are called as mesodermal cell. Now anterior end of primitive groove, this is the anterior end of primitive groove, communicate with yolk sac cavity present in endoderm. So anterior end of primitive groove communicate with yolk sac by an aperture called as blastopore. So this aperture is future anus. You know, anus is alimentary canal, last part from waste material is emerged out. So future anus is developed from blastopore, seen by anterior end of primitive groove communicate with yolk sac. Now the embryonal disc now has differentiated into three layers: ectoderm, mesoderm and endoderm. Senior diagrammatic representation in detail. This is the morula, no doubt solid stage, solid multicellular embryo like structure is converted into blastocyst. Seen here trophoblast cells, inner cell mass embryoblast cells, and this one is blastocell cavity. So from morula develop blastocyst, and from blastocyst develop the previous structure of gastrula have first developed endoderm layer, then developed ectoderm layer, and from this primitive structure of gastrula is converted into a complete structure of gastrula. Seen all three germ layers like this one is endoderm, inside here seen mesoderm, and this is the ectoderm. So a complete structure of gastrula, seen all these three germ layer like ectoderm, mesoderm and endoderm. Now this is the complete structure of gastrula known as VS of late gastrula. Seen here all these three germ layers. This one is ectoderm, labeled them ectoderm, inside it cavity called as yolk sac, and this layer called as ectoderm, inside it cavity called as amniotic cavity. In between these two layers mesoderm seen, and in the mesoderm present cavity known as coelom, on the coelom covering membrane called as chorion. So here we labeled different parts: VS of late gastrula, how form gastrula from blastocyst. Up to this we discuss. Hope that you understood everything. The further process after gastrulation is called as organogenesis, means organ formation. Here organo means organ and genesis means formation. So organ formation takes place from germ layers like ectoderm, mesoderm and endoderm. Different layer develop different body organs. We details in which layer form which organ of the body seen in detail. First one is ectoderm, give rise to epidermis of skin, hairs, nails, sweat glands. You know, sweat glands are present on the body surface, secrete sweat. So sweat gland, nail, hair, skin, epidermis developed or origin from ectoderm. Salivary gland present in mouth cavity. Mammary gland, you know mammary gland, it is a female reproductive accessory gland, secrete milk. So this two is also origin from ectoderm. Lacrimal gland present in eye. Sebaceous gland present in skin, also origin from ectoderm. Cornea, lens, retina, conjunctiva, all these the part of eye is also developed from ectoderm. Nasal epithelium, detection of smell, enamel of teeth is developed from ectoderm. Internal and external ear, you know internal external ear, these are also developed from ectoderm. Foregut, hindgut developed from ectoderm. Adrenal medulla, anterior and posterior pituitary gland is developed from ectoderm. Adrenal medulla, anterior posterior pituitary gland, you know pituitary gland is master endocrine gland, secrete most important hormones. So this gland, anterior posterior part, is also origin from ectoderm. Pineal gland is also endocrine gland, developed from ectoderm. And entire nervous system means our brain, whole nervous system is also developed from ectoderm. So always remember all these part are origin from ectoderm germ layer. Now mesoderm form all type of muscles in our body except iris muscles and ciliary muscles of the eye, which are originate ectoderm. Noted this point is most important. All type of muscles is origin from mesoderm except iris muscles, ciliary muscles of eye, that's originate from ectoderm. That's originate from ectoderm. Mesoderm give rise to connective tissue, dermis of the skin, also adrenal cortex, heart, you know heart is most important organ, is also mesodermal in origin. Blood vessels, lymphatic vessels is also mesodermal in origin. Middle ear, dentine of teeth is also mesoderm origin. Urinary and reproductive duct, we detailed discuss male reproductive duct, female reproductive duct. So urinary duct, ureter, all reproductive ducts is the origin from mesoderm. Also gonads, you know gonads, male gonad called as testes, female gonad called as ovaries, origin from mesoderm. Kidney, it is also one of the most important part of excretory system, is mesodermal in origin. sclera and choroids of the eye part is also mesodermal origin. Also remember, all this part is origin from mesoderm here, except iris muscles, ciliary muscles of the eye, which are originate from ectoderm, but all other than muscles are origin from mesoderm. Now endoderm develop into epithelium of midgut, glands of stomach and intestine. Inside stomach different glands, inside intestine intestinal glands. So glands, stomach and intestine develop from endoderm. Tongue, tonsils are also developed from endoderm. Lung, trachea, bronchi, larynx. So larynx is also called as voice box, and these are the part of excretory system. Is also developed, respiratory system is also developed from endoderm. Urinary bladder, excretory system organ, temporary storage of urine develop endoderm. Vagina, liver, pancreas. Vagina we detail discuss. Liver and pancreas are digestive glands. Pancreas is also a most important gland, exocrine part as well as endocrine part seen inside it. So liver, pancreas, vagina is also endoderm origin. Thyroid gland, parathyroid gland, thymus gland, all these are the endocrine glands, endodermal in origin. Eustachian tube of middle ear is also endoderm origin. Epithelium of urethra lining is also endodermal origin. Remember, all this part of the body origin from endoderm. Here we detail discuss which part develop from which germ layers. Some part develop from ectoderm, some part develop from mesoderm, and some part develop from endoderm. Now we detail discuss stem cell. Do you know stem cells? These are undifferentiated somatic cells. Actual stem cells are undifferentiated somatic cells of multicellular organisms. They capable of giving rise to many more cells of same type, or they can also differentiate into other type of cells. Means each stem cell have capacity giving rise many more type of cells, or same type of cell, or different type of cell is also. Best example of stem cells are bone marrow cells. Bone marrow cells are present in bones. Blood stem cells, cord cells or umbilical cord cells are the best example of stem cells. They can used, means stem cell used in the treatment of some diseases like Parkinson disease, Alzheimer disease, diabetes and leukemia. So all these different disease here noted: Parkinson disease, Alzheimer disease, diabetes, also leukemia, treated by stem cells. Here we discuss stem cells, simply it is a undifferentiated somatic cells present multicellular organisms capable for giving many more type of cells or other type of cells. Most of stem cells can be used. Some treatments like diseases like Parkinson disease, Alzheimer disease, diabetes and leukemia. So here we detail discuss stem cells. A review today's topic. We detail learn embryonic development, different event or step involved during embryonic development from zygote up to 3 germinal layers, detail in gastrulation. Also we detail discuss stem cells. On the basis of today's lecture, some exercise question noted here. Which layer is first formed during gastrulation? Yes, endoderm is first formed during gastrulation. Define gastrulation. Formation of gastrula from blastocyst or blastula, such process called as gastrulation. What is differentiation of blastomere? Yes, blastomere differentiation result formation of 3 germ layers like ectoderm, mesoderm and endoderm form the cells of embryoblast. Now difference between hypoblast and epiblast. You know, hypoblast is pre-endodermal cell from developed endoderm, and epiblast is pre-ectodermal cells from developed ectoderm. Bilaminar embryonic disc is positioned in between amniotic sac or cavity and yolk sac. How many days after fertilization actual gastrulation occurs? Yes, 15 days. Give two differences between blastula and gastrula. Yes, blastula is developed from morula and gastrula is developed from blastula. Also, gastrula is a hollow ball like cavity contain blastocele covering trophoblast cells, inside it inner cell mass, embryoblast cells. Gastrula is seen inside it 3 germ layers, ectoderm, mesoderm and endoderm. Each layer contain cavity respectively, like in endoderm yolk sac, in ectoderm amniotic cavity and in mesoderm coelom. Now what is different between zygote and embryo? Zygote is developed after fertilization, fusion of male and female gamete. Embryo is developed from zygote, differentiated event. Which of the following mesodermal in origin? MCQ question here. Retina, enamel of teeth, heart, liver. Which one is mesodermal origin? Here correct answer is heart is mesodermal in origin. Second MCQ noted here, entire nervous system is origin from ectoderm, mesoderm, endoderm, mesoderm and endoderm. So correct answer is ectoderm, from origin entire nervous system. Label the given structure of vs of gastrula. Here seen vs of gastrula structure. This one is endoderm. It contains cavity known as yolk sac. This one is ectoderm. It contains cavity called as amniotic cavity, and between the ectoderm and endoderm, mesoderm is seen. So this is the mesoderm point. In the mesoderm present coelom cavity, and on the coelom covering chorion seen here. So here we labeled vs of late gastrula structure. Here today's lecture completed. Hope that you understood everything. Today's lecture was very much clear to you. If you like the video, then you can subscribe my BioInfo YouTube channel. Tap the bell icon and like the video. If any doubt about today's lecture, mention in comment box. Thank you.